obviously the reason why we're all here. It's the webinar on the use and abuse of AI and AI language models in cardiovascular research. This webinar is hosted by Dan Johnson from Open University and Olivia Baines from University of Birmingham. They're also the social media editors for JMCC Plus, and I'm gonna now hand over to them to introduce the rest of the speakers, and I'm sure it's going to be a wonderful webinar. Thank you.
Thank you, Davor. Hi, everyone. Thank you so much for joining us today. We've got 3 fantastic speakers lined up on using AI in different aspects of cardiovascular research. So our first speaker is Dr. Michelle Williams, who's a senior clinical lecturer and consultant radiologist at the University of Edinburgh and NHS Lothian, and she holds a British Heart Foundation funded intermediate clinical research fellowship. And Michelle is president-elect of the British Society of Cardiovascular Imaging, a member of the board of directors of the Society of Cardiovascular Computed Tomography, and lead for the online education task force for the Society of Cardiovascular Computed Tomography. And Michelle's research focuses on using state-of-the-art machine learning techniques to discover new information on in medical imaging. I'll hand over to you, Michelle.
Thanks so much, Olivia. I've just shared my screen. Shite if you can't see it, but I'm delighted to be here today. I've taken over as president of the British Society of Cardiovascular Imaging now, which is a fantastic job to have. So I'm going to talk today about how will artificial intelligence transform, or is transforming, computed tomography cardiovascular imaging. Here are my disclosures. And artificial intelligence has been around for a while, but it has been transforming and evolving and changing. In the early research, we had the first paper by Turing, the first time the word artificial intelligence was used at the Dartmouth conference. And we've had, since then, the first AI winter, which was largely driven by the fact that we didn't really have computing power or use cases or enough data to do things with this new evolving technology. Then in the 80s and 90s, we had the emergence of expert systems, World Wide Web, Deep Blue beating Kasparov at chess. But then again, another AI winter, again driven by lack of computing power primarily in this time. But since then, we've had advances in hardware, advances in software, and huge advances in model structure, which means that we're really in another huge hope and possibly even a hype cycle in AI. Where it's going next is a really good question. Is this the start of an exponential rise? Is this going to be business as usual? We have AI models in our word processing software, like we have spell checker in our word processing software. Are these just tools that we're just all going to use like we used statistical software previously? Or are we heading for another AI winter if the hype gets too much? I'm really looking forward to seeing where this goes. But there is potential for artificial intelligence, or actually more correctly termed machine learning, at every point in the patient pathway and the research pathway. And that includes doing really cool things with imaging. But from a patient point of view, it involves more simple things like finding which bus will take you to your appointment in time, scheduling your appointments on the correct day for getting the results in right time for your clinic appointment, providing management recommendations, and making sure you remember to take your prescriptions. So as a practicing doctor or radiologist, I think about how we use these things more broadly, other than just the really cool imaging stuff I'm going to talk about today. And for me, AI is incredibly important for cardiovascular research because most of the information on imaging tests is never used. And when we report a scan, we're able to put in a half page to a page of information, but there's only so much we can do in the 20 minutes maximum we get to report these scans. There's over 1 billion pixels in this CT coronary angiogram, multiple organs, multiple diseases, anything that can speed up getting the information out of that report and into the hands of doctors and patients in a more accurate and all-encompassing manner is fantastic. So I'm going to talk in the 10 minutes I've got today about how machine learning can improve cardiovascular imaging now and in the future. And I'm talking very much from a patient imaging point of view, and I'm talking very much from a cardiovascular CT point of view. But actually, these 5 things can be used across the board in all types of research, MRI, retinal imaging, preclinical, animal, cellular research, and these themes can be relevant across the board. Today, I'm illustrating them with cardiovascular CT examples, but I do other talks with MRI examples or PET examples or animal examples. So it is a lens which to see AI rather than a limitation. So the first thing is helping us to do things that we do every day, but faster and more efficiently. So I have to say, the one thing that AI machine learning tools are really good at is drawing circles, drawing round things. They were born to do it. I get very bored very quickly. My accuracy drops off very fast. I can only do about 1,000 circles before I want to go home, and I never want to draw another circle. But it turns out 1,000 circles is really good to train some machine learning models. So here are some examples with what you can do with that in CT from this group that published in Medical Physics, looking at how the AI model could be used to draw around chambers of the heart. On contrast enhanced CT, which I can do, it's just boring and time consuming. On non-contrast CT as well, though, which I can't do. So it's more than just saving me time, it's doing things that I couldn't do as well. So the first big thing that machine learning can do is giving us that gift of time. I'm very busy, so anything that gives me that is really valuable. The second thing is helping us do things that we want to do but don't have time to do. So drawing circles on non-contrast CT is one of them. The big one for me is plaque analysis. So looking at the coronary arteries. So on the left here, we've got a curved planar reformation and a 3D picture of a coronary artery in a patient with coronary artery disease. The calcified plaque is shown in yellow and the non-calcified plaque is shown in red. And this is important information which has prognostic implications, but drawing these contours manually is very time consuming. So with our colleagues in Cedars-Sinai Medical Center, Damini Day and her team, we've developed a machine learning model that does this much faster. Instead of half an hour to an hour, it does it in about a minute. And this has significant implications for patient care, because if something takes half an hour, there's just not enough time to bring that into routine clinical practice. But something that takes less than 5 minutes, well, yes, actually, this might be useful. So it's a really exciting area of giving us new things that we can do. There's also new things that we couldn't do before. There's lots of examples of this, but one I'm going to highlight today is radiomics. So this is about taking more information out of the imaging test beyond just the how bright it is, how dark it is, what shape it is. It's about taking those textural information out of the CT scan in a machine-readable format and then doing a variety of cluster analyses to take out these radiomic phenotypes of the coronary artery disease. So this is quite exciting because it's something we couldn't really do before, but it's giving us new information about atherosclerosis. Pulling all the information together is really important. It used to be a CT scan, you could say there was a narrowed blood vessel, yes or no. Now we've got lots of parameters and we need a way to combine all this information to provide patient-specific information. And this is a really important area. And then the final thing is the new developments. And this is the thing that's changed my research from changing every 3 to 5 years, getting grants around long projects, to actually, I need to be changing what I'm doing every 6 months to be staying at the cutting edge of this, because that's the sort of cycle that we're talking about with these developments. Foundation models like large language models that I'm sure you've all heard about, also generative models, and federated learning is increasingly interesting because of the large data sets that we have in disparate countries that we'd really like to bring together but can't, but could bring together the machine learning aspects. Large language models are absolutely fascinating, both in terms of what they can do and also what they can't do. I pretty much have one of these open all of the time. I'm trying out lots of them to see what they're doing and trying to work out what they've been taught with and what they're useful for. They can be incredibly convincing, but totally wrong. Particularly in specialist areas like medical imaging, they come up with absolute rubbish sometimes. But even things that you think they should know more about, I'm a big history buff, and Roman history, they come up with rubbish as well. So it's not just domain specific. These hallucinations and dementia aspects of large language models are really important to think about. But I have to say, from a researcher point of view, using it as a thesaurus, using it to help get over writer's block, these are things that are really valuable right from the start. Fortunately, it still thinks at the moment that AI is not a replacement for human expertise, and it's really a partnership thing with these models in terms of using them. They're a useful adjunct, but they're not going to replace us anytime soon. The large language image generation models are very, very interesting from a research point of view, because of not just the amazing pictures and graphics they can make, but also the information, the latent medical information that they contain. So this picture is created from this sentence, draw me a CT scan of the heart with lots of colors. And you can see it's really convincing. It knows a lot about the heart. But there are some things it's not quite right, and branching in particular seems to be a challenge across the board for these models. But there is potential here, but there are also problems. We have to be very aware of fraud, fake information, and incorrect information emerging in terms of large language models and image generation models. And we also have to think very broadly about the issues here. Biases are a hugely important thing, as well as the black box. And from a patient point of view, aspects of ethics, trust, privacy, who pays, and who's to blame when things go wrong. So humans are an essential part of this loop. But AI and machine learning is extremely exciting in cardiovascular research and beyond. So I'll stop there and hand over.
Thanks so much, Michelle. That was really interesting and a great oversight of what we can do and what we could be doing in the future. So it's really interesting to see that. What we will do is keep the questions until the end of the session. So if anybody has any questions, please put those in the Q&A, and we will come back to those when all the speakers have given their presentations, because then we'll be able to get a more rounded view of everybody's talks, and so maybe things will be approached in other people's talks that you might want to ask. So with that, it's with great pleasure to invite our second speaker for today, who will be Dr. Jeff Saucerman. So Jeff is a professor of biomedical engineering and professor of cardiovascular medicine at the University of Virginia. He completed his postdoctoral fellowship at Loyola University in Chicago, and he's now leading a research group in systems biology, cardiac systems biology, where he focuses on identifying and controlling the molecular networks involved in heart disease. So with that, Jeff, it's over to you.
Should know how to do that by now. Thanks so much, Dan and Olivia, for the opportunity to share our work. So what you're going to see here is we're going to drop down in spatial scales quite a bit, but you're also going to see hopefully some similar themes to what Michelle was talking about at a clinical scale. And for the focus of this project, what I was going to tell you about is how we're combining some of the mechanistic network information that we've been able to model in the past with machine learning for fibrosis cardiac drug discovery. And in particular, not just predicting drugs, but predicting how they work, which is not usually how AI technologies are being applied. All right, so as this, the JMCC audience is quite aware, after myocardial infarction, there's a really complex environment of wound healing, maturation, thinning and expansion of an infarct scar. And within the cells, it's very complicated as well. There are many different signaling pathways that have been implicated in fibrosis. These X's kind of mark, Burns-Blackwell's group was kind of marking like which targets they thought would be the most useful for drugs it's really unclear because it's just so complicated. And so my group has been trying to develop integrated computational experimental methods to discover not just new drugs, but how the drugs modulate cardiac fibrosis. I'll posit that the how part is like intrinsic to what we need to be doing in science. So as Michelle said, these statistical or machine learning type models have been extremely powerful at all scales. At the molecular scales, it's really common now to see these UMAP diagrams if someone did like single cell gene expression analysis. And fundamentally, these methods are really powerful in finding unbiased patterns in the data. They're very scalable to large data. You don't need to really know anything about the biology in order to run these analyses initially. However, they're more limited in the biological interpretability, and they make as Michelle was referring to, they can really make inaccurate predictions outside the training data, which is now popularly known as hallucination. In contrast, mechanistic approaches connect components to functions via like our understanding of how the system is working. And so I'll jump into a lot more detail, but on the left is the idea of like signaling networks and gene regulatory networks that explain how these things work. They can be very powerful for simulating new experiments to test that understanding. You can interpret their predictions as falsifiable hypotheses. Their predictions generalize better beyond training data in many cases than machine learning models. However, it's kind of obvious that if you're going to try to develop models of systems like this, you need to know the components involved, or many of them, and you need to kind of bring in prior information, which might be very limiting. And so we've been interested in seeing whether we can kind of build new methods that build on the strengths of these two. But starting off, I'll tell you a little bit about our computational model of the fibroblast signaling network that we published in JMCC in 2016. So this model has many of the components that are known to regulate fibroblast activation with many different cytokines and hormones, mechanical stretch at the top, and then percolating down through signaling networks down to transcription factors and expression of extracellular matrix, metalloproteases, things like this. So that would just be like a diagram that you would see, say, in a review article. And then to convert that into a predictive model, we used our logic-based differential equation modeling framework using our software NetFlux, which is very easy to use. And so if you're interested in network modeling based on that stuff, we would be happy to get you started there. And so importantly, those are just model predictions at that stage. And so we need to test how predictive those models were. And so in this paper, we identified 41 different papers from the cardiac fibroblast literature that were not used to build the network model, and then tested to what extent our model could predict the outcomes that were measured experimentally, and it hit at like 80%, showing it does like pretty well, but there's also like a fair amount that needs to still be discovered about these things. A subsequent step that we took that led, kind of motivated the current study is that we began developing methods to integrate these very basic science signaling networks with a drug bank. So that's a FDA database of drugs and their targets. So we can now run simulations on many different FDA approved drugs that we were able to predict like how they might regulate fibrosis. So kind of not going to show those details. I'm kind of showing now what we did next is, so we had a bunch of predictions about drug responses. And so we wanted to experimentally validate those. And so we used a high content microscopy system. So basically an automated system where you can do your imaging in 96 well, 384 well plates. And we had 108 treatment groups that we wanted to compare against the model. We had 13 different drugs that the model had predicted. And we tested these at 3 doses in the context of 4 different environments that are environments that are representative of what a fibroblast might experience. A model had predicted that the response to drugs would be pretty dependent on the environment of those cells. And so initially, we started by imaging DABI for the nuclei, phalloidin to look at actin, collagen, and alpha SMA, which is a common activation marker and involved in contractility of fibroblasts. So just kind of focus on like one of those drugs right here. And that is like one of the drugs predicted by the model to be anti-fibrotic is Fasudil. And so here you can just kind of showing the bare minimum here is that how TGF-beta will induce procollagen, alpha-SMA, and phalloidin, which is suppressed to various extents by Fasudil. And you can kind of see that in the representative images right here. So the TGF-beta kind of induces all three of these markers, and which is suppressed by the drug Fasudil. So that was kind of exciting to see. But looking through those images, we kind of got sidetracked by something that we didn't expect to look at. So in addition to those markers, there are a lot of things changing in these cells, aspects of their cell morphology, you see here, drastic changes in cell area, and changes in the texture and stress fiber formation within these cells. These are things that have really not been tied to pathways before, because they're usually studied for like the very specific biophysical processes. And so to cast a wider net of what these drugs were doing, even though we knew we couldn't model them at the time, we captured 137 different single cell phenotypic features. So not just the protein expression, which is what we intended to do, but also things about protein distribution and morphology and texture. And what we got from this was a matrix. So, so at the bottom here are the 108 different treatment conditions. The rows here are the 137 single cell features. And we started by looking at what we were looking at to begin with, which were these three markers. And what you can see here is that they're actually pretty well correlated. They're in this block right here of responding very similarly across different drug treatments. However, if you look beyond this kind of central block, there's a lot of diversity in fibroblast state or phenotype that is not at all captured by the canonical biomarkers for fibroblast activation. And so we were really interested in like some of them, like this one, we didn't even exactly know what this was going to be, the F-actin angular second moment. So like, well, what does that mean? So we kind of took a look exactly at some of the conditions where this angular second moment was very responsive to drugs. And so that's shown here. And we looked at like a kind of control condition and a TGF-beta condition. We have low angular second moment, which means a very high degree of stress fibers being produced, or F-actin fibers. And then on conditions of high angular second moment, it's where it's like much more uniform. So like a very uniform distribution of phalloidin. And so these are like very like stark texture differences that we weren't intending to look at initially, and are very regulated very distinctly from the expression of F-actin, for example. And so here is shown in bar graph form how TGF-beta will drop. The angular second moment, it drops the uniformity and is partially restored by a SRC inhibitor that we found just from kind of looking at that big matrix. So then I think, so you find, you know, a response in a big data set, and then I think, to me, like a big basic science question is you ask, well, how does that work? Like, how is that regulated? And we didn't really have a way of accessing that because it hadn't been delineated in the pathways. And so thinking about how we might do that, we came up with what we call logic-based mechanistic machine learning, or LogML, with the mascot of the smart camel here on the left. And the basic idea here is that at the top, this is the network model that we had developed before. They're color-coded by the different modules within the network that act in a similar way. And then, so we could predict already a bunch of things about like collagen expression or alpha SMA, but we couldn't predict something like this actin texture metric that we had measured in the stream. And so to kind of add a mapping in between that, we fit a pretty simple regression model, a ridge regression model. So there's basically a linear regression model, but it's minimizing the number of coefficients that are involved. So to kind of get like a kind of an Occam's razor, you know, it's going to be a lot easier to interpret a simpler model and not include lots of very small coefficients. And so we trained the model in that way. So we now had a way of connecting from simulations up here to predictions down here that we could not model before. And so when we did that, one of the things that Oh, Michelle made a comment about black box, which is like a challenge in machine learning in certain ways. But one benefit of the really simple approaches like ridge regression is that they're very easily interpretable. Like the coefficients that they're fitting in this case are mapping exactly how much each of the modules in the network are affecting this F-actin assembly that we had measured in the screen. So PI3 kinase and the mechanical modules are the strongest regulators. And we can now take this a step further if we run. So this is the fitting or training of that model. And so now we can apply it in the forward mode to simulate knockdowns throughout the network and how that affects the F-actin assembly. And so we can see that the strongest positive regulators discovered in this way are MPK4, and negative regulators like Rho and AKT, which is related to the facetal that I showed you earlier. So now just from that sort of screen, we're, had identified from the coupled model that PI3 kinase was a central regulator of the F-actin texture uniformity. And so we went back for another round of experiments to validate that prediction and able to show that, as we had predicted, the LY, the PI3 kinase inhibitor, increases the uniformity, but really importantly, it does not increase the expression of F-actin. It's controlling the assembly of the fibers and not their production. And it is also kind of decreasing the collagen expression. So kind of validating our ability to make this combined mechanistic and machine learning model and identify new mechanistic insights. So with that, I'd like to thank the people in my group that led this work, mainly Anders Nelson and Angela Ziegler, and several of the others that are bolded. Thank you.
Thanks, Jeff, for that really interesting presentation. It's really good to see how we can combine experimental and machine learning approaches to provide mechanistic insights and new phenotypic features as well, like you showed. So I'll now introduce our final speaker, Dr. Christopher O'Shea. Chris is a research associate at the University of Birmingham within the Institute of Cardiovascular Sciences, and has been awarded a Sir Henry Wellcome postdoctoral fellowship. And his primary research goal is to use computer sciences and physics to achieve detailed mechanistic understandings of cardiac arrhythmias. Over to you, Chris.
Yeah, thank you, Olivia, and thanks, Dan, for organising this session. I think the talks already and just a general interest in this webinar shows how important this topic is, how timely it is, and yeah, hopefully we have to add some light on my particular research interest, which is, vaguely speaking, looking at cardiac mapping approaches, and in particular, today I'll focus on how we may use, are using, and how we can use in the future AI to help us in this field. Probably don't need to spend too much time on this audience, but I thought just before diving into the AI side of it, I'll just go over what I mean by cardiac mapping. So cardiac mapping, as the name suggests, is a way we can get detailed spatial information about cardiac electrophysiology. We can do this by a number of lab-based techniques and a number of clinical techniques. I focus on two mainly, so I'll briefly spend a minute or two just talk about those techniques. The first of these, which we apply in the lab, is a technique known as cardiac optical mapping, where we load fluorescent indicators, typically for voltage or intracellular calcium, into multicellular cardiac preparations, for example, cellular monolayers all the way up to ex vivo tissues. And by imaging these preparations at high spatial and temporal resolution, we get really detailed information about, for example, here in a mouse atrium, we loaded a voltage sensitive dye, the spread of electrical excitation wave throughout the atrial tissue. And by processing and analyzing this data, which is something I'm particularly interested in, we can get really detailed information about activation, dynamics, repolarization, dynamics, arrhythmia inducibility, and so on. Now, unfortunately, and particularly if we want someone to live after the procedure, we can't really do this in humans. But luckily, we have many approaches to do this, or a similar approach in clinics. The most common approach we use is a technique known as electroanatomical mapping. So these procedures are often used to help guide procedures such as ablation procedures. As you can see, it's a very simple schematic of a left atrial ablation here. But while the ablation procedure is going on, you might also have, or even have integrated within the ablation catheter itself, a mapping catheter. The goal of this catheter is not to burn or freeze any tissue, but it's simply to get an idea of the electrical activity and the anatomy of the tissue. So during the procedure, this catheter will move throughout the tissue. At each point, it will depending on certain criteria, it will collect an electrogram. So this is a very localized, we can think of it a very localized ECG, which tells us some information about this part of the tissue. And then we can take this data and by using sophisticated processing approaches, generate detailed maps of, in this case, left atrial voltage amplitude to give us an idea of the electrical health of the heart. And as Olivia mentioned, or was mentioned briefly, I have a particular interest in how we process and analyze this data. So from the optical mapping side, we've developed softwares to analyze this data and also new approaches to get more information from the optical mapping signals. And on the electroanatomical mapping side, we have built on some of the excellent work from Stephen Williams and colleagues who've generated this OpenEP platform, which now gives us open source access to the clinical mapping data. So we can generate new tools, as sort of shown here, to look at different regional variations in, for example, left atrium or other cardiac chambers. Look for approaches that go beyond simply looking at voltage amplitude, or which I'll talk about more later in the talk, look at how we can re-annotate electrical electrogram signals that we get from these systems to better understand what's going on at a physiological level. And very similar to some of the points that Michelle mentioned when she was talking about CT imaging, there's many reasons why I believe that AI is a very important technique currently and will be for the next coming years in cardiac mapping. The first of these is that these data sets can within themselves lots of data. So if you think of the optical mapping data sets, each pixel is a action potential calcium transients. It itself is made up of thousands of data points, and there's thousands of these per image. And the same principle is exactly the same in the electroanatomical maps, that to generate, for example, these voltage maps or other maps such as activation, conduction losses, so on, there's huge amounts of electrical data at each point being collected. That doesn't mean these signals are easy to understand or process off the bat, as can be shown here by this electrogram collected during an atrial fibrillation treatment of a patient here at Birmingham. We can see that you often get this very sort of complicated fractionated morphology, and it can be very difficult to how to best analyze and interpret this data. And also, well, rather boringly, but importantly, one thing that has to be taken into account is that these data sets can often be corrupted by technical artifacts. I've used optical mapping here as an example, and which I'll talk about later on in the talk, that if you don't have successful motion uncoupling, so this is where we stop the actual physical contraction of the heart to allow successful imaging, but this is not always 100% successful, and this can really affect our signal morphology and therefore how we interpret optical mapping data. So this is good, hopefully convinces you, as I'm convinced, that AI has the potential to be a really useful tool in this cardiac mapping field. I thought next part of the talk now, I'll just focus on some early work by ourselves, which is looking to do exactly that, to use AI to help us with how we handle these data sets. So I just mentioned there the concept of how hearts in our optical mapping setups may not be fully motion uncoupled, which can create areas shown in red in these ABD and activation maps, where we get these weird signal morphologies, which effectively aren't analyzable from a physiological viewpoint. Now, currently within our software platforms and others, the way this is dealt with is a manual approach. So using the custom region of interest setting, as you see here, someone would go through, analyze the whole data set, notice these artifacts, have to go back, redraw the region of interest, taking time. There's obviously a manual process, so there's selection bias, all these things that I'm sure many people in the audience are familiar with can be detrimental to objective science. So we asked a very simple question is, can we use AI to semi-automatically find these artifacts and remove these artifacts from our analysis pipeline? And this work is actually led by Olivia here at Birmingham, where she is doing the same in the ventricular data she's recently collected. She noticed that some areas, such as what has just popped up there, hopefully the video is showing okay, where she's noticed an area of motion artifact. She's tagged the area as a motion artifact. But by doing this for just a very small number of pixels, between 10 to 20 at the moment, but we believe we can get this down to more, less number of pixels, she's able to generate semi-automatically these maps, which show areas that we don't want to analyze, for example, the background, or indeed these areas that are corrupted by motion, or indeed also show corruption by the pacing artifact that we have in the middle of the tissue, and distinguish it from the areas you do analyze, which have useful information about, in this case, the ventricular electrophysiology. And by collecting this information, she's now automatically able to distinguish in red that area that she wants to analyze from the area she wouldn't. So as I said, this is very early work, but these initial results do suggest to us that yes, we can use AI to replace the manual region of interest selection in our optical mapping datasets, including motion artifact removal. And going forward, this has many benefits and uses, for example, quicker and more objective analysis, less failed experiments where we have to get rid of the majority of the data because it's motion corrupted. And also, I haven't had time to go into detail today, but there's also a huge body of work looking at how we can use computer vision and other approaches to actually deal with these motion artifacts so we can still analyze this data. And so these kind of tools have been an excellent training tool to help apply and guide these motion correction algorithms. And so moving on to the electroanatomical mapping data systems, again, but they're very similar issues are applicable. So I've already talked about briefly how we go from these raw data sets to these sort of very detailed maps of, for example, in this case, left atrial voltage. But the fact is that currently, and the majority of the case, this will involve loading this data into black box software with some user input, for example, setting windows of interest, filtering setting, and so on, to generate these maps, which is okay, but definitely for research has two main issues. On the black box side, we don't really have full details of what is going on. And with the human data sets, human input, sorry, this is as with the motion artifacts in optical mapping, is a potential source of error. So similarly, we saw, can we use AI to automatically and more accurately annotate electrograms from these electrical anatomical mapping data sets? So what I did is built upon the OpenEP platform, built two very simple tools, not dissimilar to the tools that Olivia made, but this case applied to the clinical data sets that allowed us on the left to re-annotate signals more correctly, depending on a interpretation by an expert cardiologist, and on the right to visualize how that affects the resulting electroanatomical map. What this gives then is an ideal training set where we have currently over 80,000 electrograms, where an expert cardiologist has said, like, this is the correct window of interest, this is the correct activation time, this is a potential type technical artifact, and these are points in which it's meant to be deleted. And here you can see just in a handful of electrocardiograms where we have identified areas where we think the black box systems have effectively got the interpretation wrong, which has important consequences, both from a research point of view and also now from a clinical point of view as well. So with this data set, we're now on to which is the boring part, we're now on to the more exciting part, which is now can we develop machine learning approaches and in this case, deep learning approaches with this convolutional neural network approach to automatically reclassify and re-annotate these signals. Again, like with the motion artifacts, this works in the early stages, but when already seeing reasonable results, in this case, looking at our receiver operator curve, basically telling us whether an electrogram is suitable for analysis or not. But again, hopefully within the next coming months, we'll see much more impressive results using the machine learning approaches in these techniques. So yes, to answer that original question, just like with the optical mapping data, we believe these AI tools will be very beneficial for automated re-annotation of intracardiac electrograms, which will have better quality maps in procedure and enable more objective use of electrical anatomical mapping data for research purposes. So just to conclude, hopefully I've shown you what I believe, which is the fact that we have these large data sets with complex signals with possibility of noise and artifacts shown means that AI is ideally placed to help us in the coming years to improve cardiac mapping analyses. I've provided some examples of how we're using these approaches, for example, optical mapping motion detection and also re-annotation of electroanatomical mapping data for research. Obviously, in the short talk, we haven't got time to go into all the work we're looking at, but we're also looking at a few other areas, such as more automated processing approaches, or indeed using AI to look at drug exposure just from the EP waveforms alone, not looking at things like actual drug concentration change and so on, which again is work that we're working on to apply AI. As is the theme of this talk with use and abuse, and this is something that both speakers have already touched upon, I thought I'd just give an example of some of the caveats with the use of, in this case, large language models. And as has been already highlighted, these things are very well documented, but just a few weeks ago, I was using ChatGPT just to give myself an overview of a subject within the field of cardiac optical mapping. And I got a bit of an ego boost when I saw that it actually flagged up one of my papers as a reference. But on closer inspection, I found that this paper just doesn't exist. So this is a very real world example of one of the caveats Michelle mentioned earlier, that there is need to take everything that are outputted by these large language models or any sort of AI model with a pinch of salt, with that normal investigation you give to any sort of information that you provided. But yeah, thank you again to the organisers for inviting me to speak, and in particular all the people who helped with this work, and in particular the people in bold here, Olivia here at Birmingham for work with the motion artifacts, and colleagues over in Hamburg, so Julia, Sevilla and Kyla, for their work on the electroanatomical mapping data sets. So thank you.
Thanks, Chris. That was a really great talk and really interesting to see the aspects going from the single heart going up to the whole human in those clinical data as well, and the removal of the motion artifacts, and also really interesting to see the hallucinations that the ChatGTP seem to do with your papers. So I think with that we should open the discussion. So if you can write your questions in the Q&A box that should be on your profile. We already have a few questions in, so this actually is quite nice because it fits in with what you were mentioning about hallucinations, Chris. Maybe, yeah, we can direct that at you first, but then we can open it up to anyone else who would like to discuss that. So Charles Chung asks, in AI machine learning setup, starting with an existing model seems like a time-preserving step. So obviously, if you use something that's already there, is that a good thing? But is that more likely to hallucinate if the initial model, if that model isn't actually what was initially meant to be used for? So if you, for example, he uses the example of facial recognition for analysis of cardiac imaging. Do you see a use there? Is there the possibility to do that, or do you really need to start bottom up as opposed to top down?
Yeah, so I think, it's a great point, and I agree 100%. So with basically both points. So for example, with the convolutional neural network I showed for the electroanatomical mapping re-annotation tool, that's actually based on initially based on architectures which is designed for imaging software, imaging classifications. And I don't know the details because that side of the project's led by Hamburg. But I think that is a fundamental reason why we're seeing this okay, but not optimal performance at the moment. And I think, as suggested by the question, if we design more bottom up for specifically what we're looking at in this scenario, I think we'll get a lot better. We'll get less hallucinations, a lot better performance from these models. Because, for example, one of the things that I know some colleagues are working on, which I find really interesting, is the idea of these large language models, but for very specific tasks. So one thing I'm aware of is for medical diagnoses, so report writing. So they're actually using their reports, training their models for that, to hopefully then have this much more specialized model rather than this sort of all-purpose model. So I think that's 100% correct, that I think these more specialized models will have an important place.
Does anybody else from the panel want to?
Yeah, because a couple of other additional things on top of that. I suppose you can take the model with the existing weights, or you can take the model just as a structure and start with your new data set. And depending on the task you're looking at, they might work, or you might need to come up with something totally different. It's really important for anything involving health and research that we know the provenance of what we're using and starting with. Particularly if we're thinking about things ultimately getting into clinical care. And in terms of models, a basic UNet deep learning works really well on imaging, drawing circles across the board. It's really difficult to do better than that. But I'm sure in like 3 months' time, there'll be a totally different architecture that we're using again. So it's constantly changing and evolving, and harnessing what's come before is useful, but we have to be aware that we don't want to incorporate new biases into what we're doing. So really good question.
I see Jeff has also got his hand up.
Yeah, I think the part of the discussion was the word hallucinate. And one kind of comment I have about that word is that it seems like, so if you look at ChatGPT, it gives you a very specific prediction, like what Chris saw in his imaginary paper, right? And so like any best standard, like good practice of a modeler or machine learning should have a confidence score. There should be confidence intervals on the prediction. It should not just give a point prediction and assume high confidence. And so that to me, like this idea is really just interpolation versus extrapolation. Like, of course, we all know from Excel that if you try to fit a line through a bunch of data and project out 100 time units, it's going to do bad.
I guess that also comes back to your point that you made earlier, Jeff, that you can use the software without actually having that much biological knowledge. So it could look at, you could be looking at some data and interpret it in something that wouldn't be biologically possible. And that could be a problem, right? I guess, and that's something that needs to be considered when looking at these models. And I guess that's one of the problems with, like, say, ChatGTP, for example, that doesn't know if things are feasible or not unless it's been told before this isn't feasible, right?
Yeah, there are no safeguards. I agree. Yeah.
Yeah.
Thanks, everyone. I think we've got another question as well on can you comment with the anatomical morphological data sets? Does supervised and semi-automated training work better than unsupervised learning?
I've done both, and they both provide really interesting, different types of results. Supervised is great if I'm trying to predict death, myocardial infarction. Unsupervised is fascinating for trying to find new classifications of disease. So I would say both, great fun, really interesting, and provide different bits of information.
Yeah, I'd agree with exactly what Michelle said. So we've done both as well to look at specific points. For example, the work you're doing to look at motion artifacts, a specific thing. But we've also done quite a lot of work where we've thrown all the waveforms at an unsupervised network and just see what it comes out with to see if there's any interesting extra information that we're just not seeing. And that's generated some really nice sort of things that we just wouldn't have thought about to start with. So I think they both have their place and it's pretty question dependent, I'd say.
Yeah, in my talk, I showed just a very simple form of unsupervised, which is clustering. But that's how we found cool signals that we weren't anticipating. But once we knew that we had cool signals, that's where we took another very simple supervised approach. We knew what we wanted to predict. Yeah.
Right. We have another question in the chat there from Zam Kasiri, who asks, does AI in cardiology research, is it only limited to imaging and signaling applications? Are there any examples where it's used in other aspects of cardiovascular research?
Anywhere you can get data, more than 100 data points ideally, the more the better, but reports, blood pressures, text, structured, unstructured data, anything really is possible. Your imagination is the limit.
And one thing not covered in our talks is genomics. It's like a huge opportunity area. For example, UK Biobank is like kind of has been leading the way in the availability of that sort of data and to do whole new GWAS, genome-wide association studies and things that were not intended out of UK Biobank because the data is.
Thanks, everyone. And also, just a question on how important is the input, the data quality in terms of the machine learning performance? If you can comment on that.
Yeah, I would say from my point of view, various amount of times I've tried to use AI and then found out I've made a model that finds noise as its sort of its biggest thing, what finds worse signals and classifies them against the better signals. So I think that is a very important step. And often probably the most overlooked part is actual data curation, data quality is with all the resources out there now, the actual AI part is nowhere near as hard as it used to be. You don't have to be an expert programmer, you have to be, it's the actual getting good data, I think is you're 9/10 of the way there if you can do that.
Yeah, data quality is so important. And representation is the important thing, because there's no point having a perfect data set if in the real life we have imaging that's sometimes perfect and sometimes noisy and sometimes across the range. We need to have it being perfect for a starting point, but then ultimately representative to the data that we're going to be trying to analyze with the machine learning models. And across the board, we need to think about representation in terms of sex and geographical distribution and all of the different protected characteristics that can lead to biases emerging in machine learning models if we haven't thought about them, and also ones that we might not know yet. We need to be very aware of potential biases emerging that we couldn't have predicted with the way these things are learning. So image quality is absolutely crucial anytime you're starting with a machine learning model. But then after you've got the starting point, you need representative data, and we need to think very broadly about that. And yeah, really important to think about.
Yeah, I guess it comes back to that old concept of garbage in, garbage out, right? If you don't have decent data in the first place, your results are also not going to be that great. So it's something that probably needs to be,
I've
there
had
needs
the
to be
same
some
problem
kind
as
of, you know, way that we can make sure that the data going in is the highest quality possible, I imagine, right? And there should be some safeguarding of that as well to make sure that, you know, in these LLMs that that's the case.
I have the same problem as Chris. I've made some amazing models that predict someone's heart rate.
So we have another question in the chat here, who is maybe we kind of already answered this, but they're saying it's from an anonymous attendee. They're saying they're very new to CAD research, and is there interest in the community in clinical data and genomics data and the ability to classify CAD? So I think maybe we've already covered that mostly in terms of the fact that, you know, your imagination is basically the end destination is the only limiting factor there. So maybe we can go down to a question.
The only quick comment I'll add to that, Dan, is that
sure.
the use of polygenic risk scores, where you're looking at a very large combination of genes that may not have been any of the individual genes you would have thought of causally, can be predictive. So I think that's changing views on how you use that type of data. And it only emerges from mining very large clinical databases.
We've got a more general question here. What's your advice for people coming from a zero coding background, specifically in medical imaging and specializing in MRI? So maybe guided more towards Michelle here, but interested in pursuing postgraduate studies in AI and machine learning for cardiac MRI research.
Yeah, how to get started is a great question. The first thing I'd say is there's huge amounts of resource on the internet to give you a kickstart in getting started in these areas in terms of tutorials, step through how-to guides, beyond just the can you classify numbers as the right number, but actually there's imaging step-through guides. And a variety of different domains have these how-to tutorials. So that would be my first starting place. And then find out who locally is interested in this sort of thing. If you reach out, you'll find that there's lots of other people around who are getting started in AI, or maybe there's programmers who are interested in getting started in imaging, which is a large number of people actually. So find a friend, create a local group, and get going. There's loads of exciting research to be done in this area.
It's really great. So I think maybe more of a general question is, you know, do you think that, obviously we're talking about some clinical data here, and there's a lot of work being done in the clinical side of things. What would you say, you know, obviously there might be some issues with terms of data privacy. Where would you kind of see that? Is there ways we can mitigate that? Is there any like legislation in place at the moment to start thinking about those things yet? Or is that still too much in its infancy to be able to think about those?
It is really important that any of the data that we're using is being used under the correct legal basis with permission to use it for that reason, and that there is provenance of that data. So it is fundamental to anything we do. And it is possible to use clinical data under those rules with the correct permissions and the correct authorizations. It's a complicated process to apply for all these things and organize all these things. But in terms of large data sets in the UK, for example, UK Biobank's been mentioned, and there's other resources like that emerging. But yes, privacy and data security and patient protection and patient involvement in the research that we do are all really crucial.
Well, one thing I think about in this space, in addition to the data provenance, is like how these companies are going to be combining all this data to make their own predictions. And I think you see that in like the commercial space with all the artists. They're like pulling out from all these anime artists and everything, and they're recombining the way their art in ways that can't be directly attributed to the source. And I worry that healthcare companies or insurance companies could do that, where you can't prove where they got the data, but similar to what Michelle said about a heart rate predictor, they could predict your trajectory based on some combination of things you don't even know that they have.
I guess that's probably one of the issues I saw. I don't know how it's how it's moving forward, but I remember seeing, you know, people looking at atrial fibrillation detection using shopping trolleys, for example. You know, who owns those data, and where do those data go? Do they end up with the health insurance? Do they end up with the supermarket? You know, it's a difficult one to know where to come from.
Yeah.
I see Davor has his hand up.
Yeah, I mean, sorry, I'm going to jump in because this is so interesting. But I have one comment and one question, if you don't mind. So Fu Song, I don't know if you're familiar with some of the work that he's published recently at Imperial College. He's just presented his work at the ISHR meeting in Toulouse. And he's shown very clearly that they can predict very accurately on the basis of an ECG the mortality, the time of death, actually. So you can imagine if you're able to get that kind of information as an insurance company, this might be quite useful to you. But equally at the same time, do you want your insurance company having this data? The positive thing is that it can be changed. So, you know, if there's an improvement in health through medication, through, I don't know, for example, exercise, etc., then times do change as well. The time of death, predicted time of death changes. So I actually found that quite interesting and scary at the same time. But I get a lot of questions. The question that I have is, this is more of a comment. The question that I have for three of you who are using AI a lot, and obviously clearly using language models a lot as well. At University of Birmingham, at least, we tell our undergraduates that they are not allowed to cheat using language models. Okay. In exams, for example. Now, I have my own viewpoints on this, but from your perspective, I want to hear your perspective from all three of you, and feel free to disclose as much as you want. Would you advocate the use of language models, for example, paper writing, grant writing? You know, is this something that you think we should be doing, or do you think this is something that actually we shouldn't be doing?
I think the point Michelle made about sort of writer's block, I think that's sort of how I tend to use them at the moment, sort of as first drafts and writer blocks. But as shown by the hallucinations, other stuff, it still needs that expert eye to curate and show actually building for this product. So that's just my personal experience, a personal point, how I use them on that side for sort of papers and grants and that kind of stuff.
Yeah, I think we need to be open and honest about when we are and when we aren't using them, and expect the undergraduate and postgraduate and other people to do the same. And in terms of how we examine people, we're going to have to start changing how we examine. If you want someone to just do it based on knowledge, it needs to be closed book in a room, your mobiles left outside. But if we're examining how people use knowledge, that's a different thing. And very much for me, these models have become, I used to have a thesaurus sitting on my desk helping me with words and phrases, books of quotes on my desk to help for pithy things to start papers off with. But now I can get that information from these models. So in terms of how we're going to use them in the future, I think we need to keep a watching brief on them, and it's going to evolve. They're going to end up ultimately a tool like everything else, and we list our tools in the methods, so why not list this in the methods? But we do also have to be really aware, and I have already seen one paper sent to me to review, which was clearly someone had put one of my papers through ChatGPT, changed some of the results, so that was probably their results, but everything else about that was just a rewrite of my paper. They didn't reference my paper, and I knew it was my paper because I know these papers well. But you can see where issues of plagiarism and fraud are going to emerge in the medical literature and elsewhere. So I think if we're using them, we need to be honest about it, but we have to be really aware, not just in exams, but more broadly about where they're being used nefariously.
So, Davor, I thought I'd comment more on the educational aspect of what you described. So there's been a lot of work on writing and whether it should or should not. But so I teach a computational methods for biomedical engineering undergraduate course. And so they already know how to program, but now it's like how to like do algorithms, numerical methods, things. And so I allowed them in all the problem sets. Well, first I taught like how LLMs basically work. And then I taught them how to program using ChatGPT. And then I allowed them to optionally use ChatGPT through, I think there's 12 different assignments for just coding numerical methods. And they had to like apply it different biomedical data sets. So just real quick though, the most interesting thing was that it did not affect the mean completion time. In fact, it greatly sped up the fastest coders in the class by like threefold or so. But I would say, I think like at the slower coders in the class got slower because they were really confused going back and forth between how to debug in ChatGPT and debug their manual code. So this was a surprise to me, and actually raises questions in my own mind of whether or how I will do this in the future. Because like, who are we trying to help? Yeah.
That's
Really?
really interesting
Very
information, actually, Jeff. I wouldn't have expected that. So that's yeah, especially like from my point, from the education side, you know, because we're at the Open University, we don't have, you know, we are distance learning. So it's a big discussion with us at the moment in terms of how we handle it. And one of the things we're thinking of are using these model, you know, using a model and giving that to a student, for example, and saying, what do you think of this essay? What's wrong with it, for example, and things like that. So in teaching people how to use these models and not thinking of them as a cheating method, for example. So it's an interesting discussion. And I think we've still got a long way to go in terms of education for that as well. So it's going to be interesting to see what happens in the next years, I think.
I think we've got the final question in the chat, which is, do you think there will be independent softwares which are integrated with AI related to cardiovascular research coming out soon or made open source and with increased uptake of machine learning approaches, etc.?
Yes, I think the answer to that. Yeah.
I think Chris is working on it.
Well, yeah, but I definitely know I'm not going to be the only one. So I think over the coming years, from a research point of view, you're going to see. I think the open source revolution is going to happen, but I think as with other areas, I think AI is going to be speed that up as well. In my personal opinion, and it's going to come in loads of different areas. But my field of cardiac mapping, but I think you can apply it to X, Y, Z in terms of research tools.
I would guess Michelle's clinical space is probably the most lucrative and scalable sort of area where they don't need to know all the details, right?
There's fantastic models already available on GitHub that we use all the time for our research. And as Jeff says, from a clinical point of view, there's AI integrated into a variety of softwares already. It's just going to be more and more. It takes the boring things away, drawing circles.
Okay, guys. So we've reached the end of our hour, and we've actually gone over and it's a really interesting discussion. I think this could carry on well into the night, but I'm sure. But unfortunately, we probably should close the discussion here. I would like to thank all our speakers. So thanks to Michelle, to Jeff, and to Chris for their input and all their talks, and also this really nice, fruitful discussion that we've had, and also to Davor for organising this, and to my co-host Olivia for co-chairing this meeting, and also to everybody else for their participation. So thank you, everybody.
Thanks very much indeed.
Thanks, everyone.
Bye, everyone. Bye. Thank you.